last time you were unable to make it up.
Actually, it was a very tight schedule that time.
Yeah,
for the traveling to Vellore. But next time I'll take care about that.
Sure.
So can we start?
Start. Nirmala sir, you can start.
Sir, please share this one.
George, permission to share the screen.
Yes, done.
The next talk of this session is will be delivered by Professor Thorad. So let me introduce him. Dr. Thorad is a Marie Curie Fellow scientist working at the Department of Biomedical Engineering at the Warsaw University of Science and Technology, Poland. He is the recipient of various prestigious fellowships including JSPS, IRC, ACRV Overseas, and Marie Curie. His research is based on the area of nanobiotechnology and theranostic, especially for the use of magnetic nanomaterials for cancer treatment. And Dr. Thorad is currently serving as a management committee member of the European Commission Horizon 2020. And Dr. Thorad has published more than 45 peer-reviewed journals, research papers, and articles, 3 books, and 6 book chapters with an H index of 25. So thank you, Dr. Thorad, for accepting our invitations. And now I request Dr. Thorad to share his talk. Thank you very much. Please take over.
Thank you. Just a moment. I'm using the Zoom, so.
On the down, you can see screen share screen.
Okay, thank you. So now you can see.
Can you make it in full screen mode?
Yes.
Present.
Thank you very much, organizers and the management committee who is looking for the organizing this conference. And I'm really glad that they invited me to give a special keynote talk for this. So I will be more on the talking on the what we are doing currently and what from last 10 years, what I'm doing, which research, our group research focus and some highlighting that. So as I know, the conference is more focused on the flexible electronics. This might be something interesting for you. So I'll be going more on the first introduction and we'll be showing our research results at the end. But I'll be starting with the more introduction. So you will be involved in the concept of this nanomedicine and how material science are used to tackle the issue of health emergencies. So I'll be more on the cancer. We work on the infectious diseases as well, but today's presentation will be on the cancer. Just a moment. So we work in the European Commission project. We have the Europe, so it has a group of 28 countries. Our country, that is Poland, is somewhere in the east side of the Europe. And our institute, it is established after World War II. I'm not only associated with this institute, but I have association with 3 different institutes. First from the Poland, another is from the University of Zurich, where we are doing some animal studies. And we have a collaboration institute from the Limerick, Ireland. So I have been there for 3 years. So it is actually a big group and we work together with solving these issues. So let's start with the what European Union and how we work under this umbrella. So European Union is a part of, is a group of 28 countries. Now Britain is exiting, but still we are together. European Union is a political body, and they have the bureaucratic body called the European Commission. So all research money coming through the European Commission, and they have a special wing called the European Research Council. So they are funding all the research related through European Commission. The European Commission has their strategies for the cancer. So the cancer, they have the 4 main pillars. So first is the prevention. So try to prevent that disease. If don't, then start to diagnose. Once we diagnose, then use the treatment, and using the treatment, improve the quality of patient and the overall life survival of the patient. So these are the main 4 pillars, and this is called the European Union Cancer Plan. So every 3-4 years they are improving it is by updating with the new research and technologies. Let's start with the cancer. So I believe you belongs from the some basic science concepts or even some medical concept, but overall the cancer is just a disease that it doesn't come from outside. So like the viruses or bacteria, some bacteria, they are responsible to make a cancer, but cancer is a disease that growing inside our body. So the cancer cells are our normal body cells, but with the time they change their DNAs, there is some defects in the DNAs and they uncontrolled growth. So cancer is nothing but not only a single disease, because it has arising from our own body. So it has a group of diseases. So till now we diagnose more than 100 type of different diseases. They come together when a single patient have a cancer. So it is a big attack on our immunity, and that's why the cancer patients, their mortality is very high compared to even coronavirus. So every cancer has their own biological molecular features, pathological features as well. So I'm not going in detail with that, but just statistics about the cancer. So as we know, the cancer, it acts as like a traffic control. So when cancer cells start to growing, means the normal defective cell of our body. So they are just normal cells. So they try to grow and grow, but they don't stop. So they don't follow the traffic rule. Sometimes their growth is slow, sometimes it is very fast, but they grow very fast. And we can't control at a certain moment drugs or our immunity. It gets failed to control such a growth. And that is nothing but the tumor. So when we go to the statistic or the features of such a growth, so cancer cell, let's say, it grows very fast in the in vitro condition or in the laboratory. So we can grow cancer cells within 24 hours. But when in the human body, they grow around 2 to 6 weeks, they get a bulb, then they form the tumor. So on an average, 1 million cancer cells, they look like a pea. So if it is a billion cancer cells, they like a grape. And a normal tumor in the body, they have many billion cells. So it's a big amount of cells. They need energy, they are hungry, so they are taking the energy from the normal cells. And then we get weaker and weaker. So in men, when we divide the cancer in between men and women, so men have more on lungs cancer, they have prostate cancer, they have kidney cancer, but the lung and prostates are the main or the more they are contributing more than 30% cancer mortality. While in women, it is a lung cancer, but breast cancer is the very dominant in women. And they have cervical cancer as well. They have urinary bladder cancer. Some cancers are common in men and women, like lung cancer, even breast cancer. Some men, they get diagnosed with breast cancer, but the population or the numbers are less than the women. Well, these are the recent facts about the cancer. So how much contribution by different type of cancers in mortality or number of cases. So in general, overall, every year, around 10 million cancer patients get died. And let's say it's 10% of total diagnosis. So we can think how much diagnosis every year for cancer patients. So just I will give you the one fact, just from January to the June or May end. In the world, more than 10 million cancer patients get diagnosed, only men. So I have collected the data from the International Cancer Agency. So 10 million men get diagnosed with the cancer patients. While coronavirus, you can see it is 500,000 together till the May end. So it is, a very, very dangerous disease than the another. Expect the heart diseases, because heart diseases are still dominating the death rate and the diagnosis. But the cancer is on second, and that's why European Union, not only European Union, every country has their cancer strategies, including India, America, all even African countries, they have their strategies to tackle the cancer. Just I will show you a small video. Those who have or those don't have the background of biology or the, let's say, cancer. So how it generates in the body and how it starts in the body. Yeah, so in the United States, it is 1.4, but on an average in the world, it is 1 out of 8. So they die with the cancer. Now let's focus on what type of therapies are used. So I'm not going on the diagnosis techniques because we don't work on more on the diagnosis, but we work on the therapies. We are now more focusing from the September on the diagnosis of small pediatric patients or children. So still now we have 4 to 5 type of treatments options like surgery for brain or some accessible cancers. Radiation is used for every cancer treatment along with the chemotherapy. And they have the common protocol called the CTRT. Targeted therapy is now becoming very famous using the nanomedicine and other biomolecules. Hormone therapy, or there is another immunotherapy as well. So these therapies are very famous now, but they are very expensive and still on some lab scale research. But the surgery, radiation, and chemotherapies, the 3 are the most dominated therapies for cancer. Well, from the beginning of the 20th century, so discovery of the radioactivity and some radioactivity molecules. So the radiotherapy gets dominated in cancer. After discovery or the designing, developing of new drugs and compounds, chemotherapy get attracted by the researcher and the clinicians. And after the new era of nanotechnology and others, targeted therapies are now focusing, or researcher, clinician getting interest on the targeted therapies. But still the chemotherapy and radiotherapy is dominating the cancer therapy. So there are so many drugs already in the market for cancers, and it depends on what type of cancer is there, what type of, and they use the protocol. So clinicians, they don't call the drug, they call the protocol, because they combine many drugs together now. Earlier they used only one drug, but now they are combining many drugs. Well, these are the therapies that I already mentioned: surgery, radiation, immunotherapy, and targeted therapy are the now growing interesting. Stem cell therapy as well, nanoparticle delivery, some lipids, some liposomes are already in the market, so they use for the cancer therapy. In on average, like surgery, it is just a removal of the particular tumor cells, and their tissues. Chemotherapy is just healing these tumor tissues using the drugs. Radiotherapy is more focused on the using the external radiations. They are high energetic, so they are dangerous for the normal cells. So focusing and targeting a specific tumor. Now very sophisticated instruments were there, so it is not difficult to target the radiation therapy, but the still output is low for that. Well, what we are now doing, how we are getting support from the European Commission to focus on the new cancer research. As I mentioned, the European Commission is a non-bureaucratic or bureaucratic body of the European Union. So this is called like all officers, scientists, they come together in Brussels. So they have the main office in the Brussels, Belgium. And I have been associated with some European Commission cost actions. So cost actions are the cooperation in science and technology. So we have group, especially scientists, those are working in the cancer, or those are working in the nanomedicine, or those are working in the non-conventional cancer therapy. So I am associated with the 3 cost actions, and we have around more than 350 research scientists from the Europe. So we yearly once we meet somewhere in the Brussels or somewhere in the Europe to design the new strategies for cancer, to design the new ways, or discuss outreach activities, give some talks to common people. Or more focus on the nanomedicine. Nanomedicine is a new technology that uses the nanotechnology in the medicine. It is called the therapeutic branch of the nanotechnology. So it deals with the designing new type of hybrid materials, organic materials, inorganic materials, biological materials as well. So mostly we work on the inorganic materials and uses the organic materials to make a hybrid nanostructures, hybrid nanoplatforms, let's say. So when we compare with this nanotechnology with the biological entity, so all we know now, we are more on the curious about the viruses and bacteria. So when we compare our nanotechnology, it deals in between 1 to 100 nanometers. So in that range, some gold nanoparticles, some inorganic material, carbon nanomaterials, they comes together. Above that, some liposomes. So these are more on the organic materials. So they used to deliver the drugs. So they belongs in, when we compare these liposomes, they are like the viruses. Viruses has the size in between 100 to 500 nanometer. Even the coronavirus is around 150 nanometer in size. Some proteins, they have the very small structure in between 10 to 100 nanometer, but proteins have in between 10 to 50 nanometers. So some viruses like coronavirus, they have the sealed of this protein. Our blood cells are bigger, so they are in micrometer range. While we don't call any material who is in micrometer as a nano. So in between, the material in between 10 to 100 is termed as the nanomaterial. Well, there are different types of materials, organic materials, inorganic materials, and combining organic, inorganic. So we are on interface of making a inorganic and organic materials. So we call nano hybrid platforms. So we use the gold, we use the magnetic material, we use the silica, silver as well for cancer and infectious diseases. So, till now, these are the different materials are used by various researchers for the cancer therapies. Not only material, we use the physics of the physics for the therapy as well. So when we differentiate the any matter in the our body, we can differentiate in between physics, chemistry, or biology. So let's get an example for biology. So how we can call all our bodies a biological matter. So all our cells, they have the intrinsic part of DNAs, and they have the molecules. So all biological things. So all our cells, including from our hairs to the our nails. So they all are some cells. They are proteins or they are DNA. So this we term as biology. So the whole body is just biology. Let's think about the chemistry. So how we can term this all our body is just a chemistry. So chemistry, it deals with the elements, it deals with the molecules, it deals with the atoms. So all our DNAs, all our proteins, they are, carbons, they constitute carbon, nitrogen, hydrogen, oxygen. So they are all elements. So this is all about the chemistry. Well, very interesting, you will know when we term our body as a physics. So how we can call a whole body or matter is a physics. So physics deals with the energy. So when we term our whole body is a physics, means it is an energy. So our body, that DNAs, that all molecules, proteins, they bind together with some energy. They need energy to bind together for molecules to transfer from one part up to another part, like blood circulation, to go from brain to blood, or from heart to liver. Everywhere they need energy. So our body is just energy. And we are using, in the universe, we have mainly 5 energies that we are utilizing for our daily purpose, including light, magnetic, electric. Electric, we call it is a synthetic type of energy, but magnetic, light, these are natural sources. So we are more dealing on the light and magnetic, and we call it is a therapy, magnetic therapy and light therapy. We use the same kind of nanomaterials to excite these materials under the light. Or under the magnetic field. So magnetic materials, they are responsive to the magnetic energy. Light materials like carbon, gold or silver, graphene, they are very excited to, they are very responsive to the light energies. And we are combining both. Well, what we are designing, we are actually working on the nanomedicine for the designing new therapies. So these are non-conventional. So they are based on the magnetic things, or they are based on the electric energies. So we call it as a hyperthermia. So hyperthermia, hyper means it is increasing the energy or temperature. Thermia is nothing but the temperature. So I will come to the small video so you will understand what is the hyperthermia. But hyperthermia is nothing but the increasing the temperature of any system for a certain time, and utilize that temperature to kill the tumor cells. So tumor cells, they are very sensitive to the temperature, and in between 44 to 46, we can kill tumor cells. But some normal cells of our body, they can survive. It's not for a long time. We can use it, this kind of temperature, very short time, let's say for 10 minutes, 20 minutes, or half an hour. And this must be a localized. So we can't keep our whole body in this temperature for long. So we can keep only organ or the infected cancer organs like tumors for short time. Well, these therapies, they are targeted and not targeted as well. So non-targeting is one of the drawback of nanomedicine, and that's why we are working on the targeted thing. We are using the light to target this and magnetic as well. Both energies we are using to target this. Well, in the light, we know light consists with the three intrinsic part, UV and visible and near infrared. UV is very high energetic and it is dangerous for our body. But now we are using UV to clean the bacterial infection or the viruses as well, because they are high energetic and they damage the DNA of these molecules. When they damage the DNA of our normal cells, and that results into the cancer. So we don't use the UV. More focus is on the visible and near infrared. So we are using both spectrum of the light for exciting or stimulating the nanomaterials. But their penetration depth is less. So for visible, it is very low. NIR very high. So we are more focused on the NIR light. Well, when we interact with the light to the materials, so there will be two phenomena. One is called the photodynamic phenomena. Photodynamic means, photo means the light, dynamic is in the motion. So when we excite any material or we trigger the material under the light, so they generate some reactions. So these reactions make sometime radicals, some chemicals, and these chemicals can kill the bacteria, can kill the viruses, and can kill the tumor cells as well. So this is nothing but the photodynamic therapy. Well, if any material under the light and if it generates the temperature, then it is called the photothermal effect. And that photothermal effect is utilized to kill the tumor cells using the hyperthermia treatment. So this is all about the light. When we use the magnetic field and use the magnetic material, then it is called the magnetic hyperthermia. So this magnetic material, they excite, they generate the energy under the fluctuation of such a magnetic field. And this energy is nothing but the magnetic hyperthermia energy, and that we utilize to kill the tumor cells. So here you will see a small video about how magnetic hyperthermia works. So this is, let's say, a human body. We have to design some magnetic field or the generator, magnetic field generator, who will generate the magnetic field. Here is the tumor. This is the magnetic field generator. It is like a MRI machine. And it would be a future technology where the magnetic field will be generated. Already MRI, they generate such a magnetic field. So when this magnetic field, we use the magnetic material under such a magnetic field, they generate the heat from our normal body temperature to, let's say, 45 temperature. And in this temperature, we can kill the tumor cells. So this is a new technology. We are working on designing such parameters as well in our laboratory with our colleagues. So human body with a tumor, we can kill the tumor, we can vanish the tumor, eliminate the tumor from our body. So this looks a simple technology, but it requires too much efforts, and still we are on the designing technological things. Now I will come to our research. I'll not taking a long time to go in detail of our research, but because we have some technological advancement, but we have some IPR rights as well. So I'll be just explaining how we are designing the new nano platforms that will be used for such a novel technology in coming days. So we are working on different materials. So here example is the graphene. Graphene, which is very, very interesting material because it has a high surface area, it has a large amount of surface available for drug conjugation, for functionalization, and it has a light absorbing properties. It doesn't have the magnetic responsiveness. So we used the graphene oxide, and we combined it with the magnetic material, so it can be active as a magnetic material. So overall, the graphene we functionalized with the iron oxide. So now it is a kind of super paramagnetic graphene oxide material. Super paramagnetic means it is showing the magnetic properties only under the magnetic field. Then we use some polymers to conjugate the biomolecules, including drugs, including aptamers. Aptamers are the cancer targeting agents. So they only target cancerous cells. So we can get the targeted therapy. And we made some hybrid nano platform with the drug, aptamer, and inorganic graphene, magnetic material. So it looks like this. So it is a TM image of graphene oxide, and this is conjugated graphene with the magnetic material. We have the graphene with magnetic and gold as well. So we are working on some imaging of that material. So this material, when we did the surface elemental mapping, we got good magnetic coverage of this graphene surface. So it is very good magnetic properties as well. So we detected the concentration of iron oxide and graphene under the XPS. But very interestingly, we found this material as a super paramagnetic as well. So it has a magnetic property. The graphene has a paramagnetic material. So it doesn't have its own magnetic responsiveness, but when we conjugate with the iron oxide, it has a magnetic responsiveness. And this we are utilizing for the magnetic hyperthermia and light absorption. So we made a graphene magnetic nanofluid. So under the magnetic field, it is showing the magnetic responsiveness. And we are using now for the animal trials in the jury where we have the setup for the animal experiments. So these we are using this induction coil which generate the magnetic field and magnetic hyperthermia. Not only a magnetic hyperthermia, but we are using the light as well. So under the magnetic field, these materials showing the two poles, yes and N, as they get excited and magnetic field. So here you will see at the end, all the material get influenced by the magnetic field. So this is a graphene magnetic nanofluid. Well, without the magnetic conjugation, this material is not responsive with the magnetic field. Well, so this is the system that we are using for the magnetic hyperthermia. So we have another system conjugating. We are now combining magnetic and light system together. So this is our design magnetic system. This is our system that we use to activate the same nano platform, hybrid nano platform under the light, laser light. So we designed this small electronic device, laser device for the laser activated drug delivery. So it has the laser and the thermo camera. So this is just combining electronic engineering with the computer engineering. So this device we are now coupling with the magnetic device. So well, what we did, so we have combined this nano platform with magnetic and light platform, light, and we found that this material, they are combining drug and they are releasing drug as well. So this is a fluorescent spectra of the drug combined magnetic material. So doxorubicin, that is one cancer drug we use to functionalize this hybrid nano platform. So it is showing the fluorescence. And here you will see with respect to the time and the pH, the drug is released. So this is our cancer cells, colon cancer cells we use. We are using breast cancer cells as well and brain cancer cells. So you will see with respect to the time after 24 hours. So this drug is released into the cellular environment and around the nucleus, but some drug is even went to the internalized into the nucleus as well. We did some magnetic field dependent and light field dependent drug release, and we found that under the magnetic and light field we can improve the drug release So this is in the pH of the tumor. So tumor pH is around 5, while our normal body pH or the physiological pH is 7. So in tumor pH, we get a more drug release. And at the next experiment, we carried the magnetic hyperthermia on these tumor cells, and we found almost after 30 minutes of temperature, we almost killed 98% of cancer cells. So these green cells, they look live cells, and these red cells, so they are stained with the propylium iodide, which stains only the red cells. The dead cells, and it shows the red fluorescence. So with respect to the time of the therapy, we can see the more and more red cells. So the cells killing rate is higher. Well, this is the part that we are doing. We are doing more on the technological part. Like we are designing very small instrument and the coupling fiber, so we can go into the liver tumor, we can go into the breast tumor, we can go into the brain tumor as well. So we are trying to that as well. So we have designed very small optical fiber. Unfortunately, due to we are preparing the patent for that, so I didn't include this data in current presentation. But overall, we are designing the hybrid nano platform that is active under magnetic and light field. And it is showing the very good in vitro results. We have in vivo data as well. So we are not only collaborating with the institutes from the Europe, but we have very good collaboration from India as well. So we are doing some animal experiments in India, like Manipal University, where we have good animal setup. Okay, this is our project. That is the project that financed by the European Union Commission, Marie Curie, and we have the next stage of this project that will be implemented in the University of Oxford from the September this year. So this is a nano cargo that is more on the focused on the designing the magnetic and phototherapies, but the next project will be more on the designing instrument technology for the brain cancers. So we are not only doing the research and publishing paper, but we are doing the outreach and dissemination to go to the normal or the common people. So we are publishing books. So we have published one book recently with the Institute of Physics, UK, and another very recently come into the market by elsewhere. So it is more on the breast cancer. We are now publishing 2 more books on the brain cancer and normal pharmacokinetics. So this is my last slide, which is related with the, not related with the research, but it is the common sense that we need now after the coronavirus outbreak. Now research is more focusing on the health domain, and we are requiring the basic scientists, we require the clinician, they come together, doctors. Unfortunately, this is not a scenario in India or worldwide, but it is good scenario in Germany, where I worked for 1 year with some hospital. So this, the lady, she's a German president. So once she said in her presentation that we need a scientific minds to tackle the health issues, not only health, but all challenges, environmental and everything. So she herself is a PhD in quantum chemistry, and the Germany is the best example how they tackle the coronavirus, because of they made the task force with scientists, with economical scientists, clinician, and epidemiologist, everything. So we are very neighbor now, and even I frequently travel to German institutes. So those who are today in the presentation, those who belong from the computer or all other fields, I would suggest them, I would request them to bring together your ideas and challenge the current issues, not only health, but I encourage the energy, I encourage the environment issues, economical issues, and we need such an interdisciplinary mind. I have a basic degree from physics, but totally I'm now shifted into the biology, and we are doing the animal studies. I'm doing the inducing tumor in the mice, inducing tumor in the rat, and rabbit. So it is a whole transformation from basic scientist to the clinician. So we need such kind of human resources to tackle the health issues. So thank you very much for your kind attention, and I would like to answer your questions. Thank you.
Dr. Neeraj, can you unmute?
Yeah. Thank you, Professor Thorar, for an informative talk. So there are a few questions raised by the participants. So I may ask, I may pick up a few questions so that you can answer. Can you hear me?
Yes, yes.
Yeah. So the first question is, during magnetic hyperthermia or photothermal therapy, how can we control the temperature in human body?
Yeah, that is the one problem not only for the magnetic hyperthermia, but all photothermal therapy. So control of temperature within the body. We can control by using the two different phenomena. We need the monitoring. So the one issue, the hypothesis is that if we make some magnetic material, which changes their phase from ferromagnetic to the paramagnetic, when we reach the temperature around 45 or 46 degrees centigrade. So let's suppose a material who has generated the temperature around 46 and change the ferromagnetic to the paramagnetic, then it will stop to generate the heat and the heat will remain there. So this will be the solution given by the physicist, but it is not accepted by the biologist and the clinician because this is just a hypothesis and controlling within the tumor or within cell difficult. So that's why we have recently designed the optical fiber technique. So we can go into the tumor and excite this material into the tumor and use the IR thermography. So I shown the one image, we have the IR camera. So we are now using this IR camera. We are designing the IR thermography, which can sense the tumor temperature from the outside. So this can be the another solution in future.
Okay, thank you. One more question has been raised by Gokara Konda, one of the participants. What happened to nanoparticles that enclose the medicine after it release, and do residual nanoparticles pose health risks? Are there any research study on those aspects?
Yes. This is called the nanopharmacokinetics. So when the nanoparticle injected to the body, let's forget about the humans because we are not on the human trial. We are all doing on the animals, only mice and rats. Even not we are doing on the rabbits or the sheep or the dogs because they are more familiar with the human. But when they enter into the body, they travel they go into the liver, they go into the heart. It's very difficult to reach the brain, but they, kidney filters it. So this is called the nanopharmacokinetics. So when we enter, let's say 100 nanoparticles, we probably get 2 or 3 nanoparticles into the tumor. So now this is another part, nanopharmacokinetics. So we can reach more and more particles only to the tumor. After the therapy, they probably into the tumor, or when we remove that tumor, they go with the tumor, but probably they live in the tumor or probably in the liver, but it depends again on the size. If they are very small in size, they can filter it out by the kidney as well. But this is the issue with they are remaining in the organs for a long, and they are again making a toxicity, organ failure. So this is another part of the research domain called the nanopharmacokinetics, and we are working on that as well.
Yeah, thank you. One more question by Satish Kumar. How these nano formulations can be administered to cancer patients?
Oh yeah, it can be like our normal drugs, maybe orally, maybe intravenously, but more focus on the intravenous. And what researchers now doing? So we are, inducing tumor to the mice, and it is on the surface of the mice, surface tumors we call. So we can directly inject these particles into the tumor. This is when we do experiments in the lab. But what about the human? Because we can't directly inject the any drug into the tumor. So we have to through intravenous or the oral. There is one interesting phenomena called the enhanced permeability and retention effect. So this tumor, they have very hungry, they have the hypoxia condition. So they can, they can attract more and more particles, and more particles can be delivered to the tumor. So this is called the EPR effect. So this is one of the hypotheses as well. So scientists believe that through EPR effect, we can target more particles to the tumor. But we are using the magnetic field gradients as well to target the tumor. So when the materials, they have the magnetic field and use the external magnetic field. So with respect to the magnetic field, we can, from any organ to another organ, we can transfer that. So this research has encouraging results on the animals, and in the future, it will be on the human as well.
One more question is there, after 30 minutes of continuous exposure to the magnetic field at a temperature of around 45 degrees, there are chances of death of normal cells in that region of treatment. How will you tackle this?
Yeah, that's called the non-targeted or non-localized therapy. But just imagine when we target this material only to the tumor. Yeah, let's say a brain tumor or tumor which we have access, like a breast tumor, we have access from the surface as well. And when we inject this material and use the magnetic field only within the tumor region, we can, find the tumor region using the MRI or CT scan, and then use this therapeutic to the only tumor, like radiotherapy. So radiotherapy or radiation, we are using only to the tumor side, not to the normal cell. Yes, there is a chances when we use this elevated temperature for long to the normal cells, and definitely normal cells will die. So this is another issue with the transforming this technology to the in the market or something. So we are organizing one very interesting workshop next week, where industrial partners who design the imaging technology, how to imaging the tumor with nanomaterials. And we are now industry and academic workshops, so how we can tackle these issues. And we are inviting some clinicians as well, because we need this interdisciplinary team to tackle this issue.
Thank you, Professor Thorad. As we are running out of time, probably.
Okay, thank you.
Thank you for your nice talk. Over to Nirmala, ma'am.
Yeah, thank you, Dr. Thorat, once again for accepting our invitation. It was really an interesting talk, and participants were really enjoying your talk. So on behalf of CNR, thank you once again for joining the conference. Thank you.
Thank you.
Yeah.